Hi Nat5, so we're still looking at proteins and enzymes in cell biology, and now we're looking at enzyme function. So before we go any further, we actually have to kind of go back a stage to stuff that you did a few years ago. Okay, so we're looking at what a catalyst is, because enzymes are a very specific set of catalysts. So a catalyst is something that, and you should know this, speeds up a reaction. That's really important. Is not used up, also really important. And also essential, lowers the energies needed for a reaction. Now that means that you can get reactions to run at a much lower temperature, and for us that means the difference between being able to run a reaction at biological temperatures and being dead. Okay, so I like this little example. Okay, if you've got a big high energy barrier then and you want to get dry frogs to wet frogs, it's difficult to get over the big wall, but it's easy to get over the little wall. So what's an enzyme then? So an enzyme is a biological catalyst. So biological meaning that it comes from something that's living, and catalyst, all the things we just said. So speeds up a reaction, isn't used up, lowers the energy you need. So the biological bit means that we're making them inside our cells. And because obviously we're looking at proteins, you should be getting that they're going to be made out of protein. So if they're made out of protein, remember proteins have all got specific shapes, and that is really how enzymes do what they do. So for every single reaction, you have an enzyme that will make that reaction happen, okay? And that enzyme will only work for that particular reaction. Because what you have in an enzyme, and this is a space-filling model of a particular enzyme in blue, the little red bit is the thing that's actually being worked on by the enzyme. So it has a particular shape that will fit it, and that shape is called the active site. So the substrate, the thing you're going to actually work on, is fitting into the active site. Right, so here's our theory of how the whole thing works. So we know that enzymes only work on specific substrates. They do that because they only have a shape that fits the specific substrate that they're working on. So our theory that we use is lock and key, which you can see, I hope, makes sense. So we've got a particular shape and only one key will fit the lock. So our lock that we're bringing in is the enzyme. So if I was to look at the active site in this enzyme, I'd be looking at this space, this kind of shape in here, okay? And that shape has to have a complementary shape in the substrate. Substrate then fits in and we get our enzyme substrate complex. And now the enzyme does its job. And in this case, what's going to happen is it's going to break the substrate down into two products. Okay, now the products no longer fit the shape because they're like half the shape, so the products get released. So our theory is that the enzyme is the lock and the substrate is the key, and you only have one key for each lock. So that bit where it fits is called the active site, and the term we're needing to use here is complementary to the substrate. So if the active site is blocked, or if we wreck it somehow, if we change the shape of it, then the enzyme will stop working. Now, if enzymes are proteins, that's something that we need to talk about in terms of anything that affects a protein will affect an enzyme. Okay, but blocking it could be something like an inhibitor. So you can actually have things that just come in and literally block your substrate from getting in. Or you can have things which stick to the enzyme and change the shape. So this has now got a kind of loop out to the side as opposed to going straight up. Okay, and this could be really quite nasty. This would be things like lead poisoning and stuff that cause that. So looking at enzymes, you need to have a kind of general equation that you're running. So the general equation goes substrate, so whatever goes in, and then over the top of the arrow, and that's important because the enzyme is not really going in and then being used up. That's the whole point of what catalysts do. So we put them above the arrow to show that they are not being used up, and finally we get our product coming out the other side. And that's your general setup. So when we look at the enzyme reactions that you have in your cell, we can actually split them into two groups. Okay, we can say that they are degradation reactions, which means that we're taking something big and breaking it into something smaller. Or we can have synthesis reactions where we take something smaller and make it into something big. Okay, and that pretty much covers everything that's happening in the cell. So we've got degradation and synthesis. So there are a few examples you're going to need to know. This is your first degradation one. Hydrogen peroxide with catalase as the enzyme produces oxygen and water. Now this is a really nasty chemical for us, and we make lots of it, but we need to break it down because otherwise it would be toxic inside our cells. So our livers actually have quite a high level of catalase because we're breaking down the extra hydrogen peroxide that's being produced. And oxygen and water are obviously not damaging. What's really nice about this as a system is that we can measure the oxygen, so it gets used for a lot of experiments. So first of all, though, you have to work out how to remember it. So here's your possible ways. HP cow, just take the initials and learn that. Brown sauce, HP, and burgers, because that comes from cows, if that helps. And obviously that's why I have a massive stuffed cow in the lab. Number 2, proteins, pepsin, polypeptides. And to be honest, for me, that's a kind of rhythm that I have in my head whenever I say it. I can't even help myself from saying polypeptides with a kind of rhythm to it. So this one, PPP, it is one of the main enzymes in your stomach. The reason it's a really useful one to know is because it's a bit weird about where it actually functions. Your stomach is a pretty hostile place with acid and stuff in there. So as I say, PPP, and this will make sense to my lot, but I have a parrot in my lab for that one. Number 3, starch, amylase, maltose. And I think that just comes out as SAM. Okay, this is one of the major breakdown reactions inside your gut, and in multicellular I go through the list of all the kind of degradation ones that are happening inside your gut. Any of those are good examples, but this one is a really nice one. We can measure this by measuring how much of the sugar is produced using Benedict's reagent. Okay, your synthesis example, of which you only have one, okay, that we're using a lot. Glucose 1-phosphate, potato phosphorylase, starch. Now that potato bit you don't need to have, but you do need to have phosphorylase, and it has to be the glucose 1-phosphate, not just glucose. So any plant that stores glucose and stores it as starch will have an enzyme that they're going to use to convert it. Okay, how to remember it though is a little bit tricky, because that one doesn't fit nicely to any of the mnemonics that I have. But GIPS I can kind of get, and GIPS sounds like chips, and chips comes from potatoes, and potatoes have lots of starch. It's a possible way to do it. You got to find a way and make sure you know them. Okay, and that's our enzymes.